Susan, thanks so much for the great introduction. I think I have a few questions that our audience will be interested in, and you can help us with those. So one of the things is you showed this great diversity of cyanobacteria, that they grow, in the Arctic and they grow in thermal hot springs. How does that diversity help when we think about biofuel production or production of anything using cyanobacteria?
Whenever you have diversity of either the cell physiology, the ecology, where it lives, what that means is that there are different enzymes there to make different products, because an organism has to be adapted well to its environment. And also the morphology is going to be different. So a cell that is filamentous is going to be expressing genes that you don't have well, a strain that's filamentous is going to be, its cells are going to be expressing genes that you don't have in some of the unicellular species. But if you think about it, an organism that lives in, for example, very high salt conditions is going to have enzymes that have evolved to work in that environment. And as we start sampling different environments, we're also just discovering new genes. So genes that we didn't even know about, and that means chemistries and enzymes that we didn't already know about. And so whenever we talk about how important the diversity is, that's because we know that means a diversity of raw material that we can work with to engineer.
So a genetic diversity as well as a physiological diversity.
Exactly.
So then you talked also about having a model system versus having a production strain. In your mind, what are the biggest differences between those two?
Probably the biggest difference is that the model organisms were chosen, in many cases decades ago, for particular properties that were of interest to the scientists who were bringing them into the lab and starting to work with them. And decades ago, we weren't thinking about how well something grows out in a pond to be useful as a biofuel strain. Very often the organisms were chosen because they did something that the scientists wanted to study. For example, to differentiate specialized cells to fix nitrogen, or because they were easy to work with genetically. And traits, they're different traits than aquaculture traits and large-scale growth traits. And so if we think about what we want to do now, it's important to find organisms that are going to work really well at production scale and then make models out of those. And what we mean by model is a strain that there is enough agreement and enough effort that goes in to work with that strain and to understand it well. And if we go back really decades to when people were studying cyanobacteria, every paper was on some different strain. And so that was great because the diversity was sampled, but there wasn't enough effort to make real progress on any one strain. And then the models went the other direction to where we really kind of know a lot about a few strains, and now we say, but wait a minute, those aren't the ones that have the properties we want to focus on for biofuels.
That's right. So just like agriculture, we'll pick a few cyanobacterial strains that will grow at very large.
Exactly.
So one of the things you talked about was this genetic diversity. So if we look at a cyanobacteria genome, how big and complex are they, and how are we going to introduce genes into the production strains that we want?
They really vary a lot. So the smallest genomes are about 2 million base pairs, and that's smaller than the genome of E. coli, which is the most commonly genetically engineered strain. And then there are others that are up around 6, 8, or even 10 million base pairs, which is a much bigger genome. And you can't really say that a bigger genome means this or that, except that typically for the strains that have a greater metabolic diversity, they are more unusual metabolic capabilities and more tricks that they can do, they're going to have bigger genomes. Now, in terms of introducing genes, it's not just that if you have a bigger genome to begin with, you can introduce more genes. The small organisms can, in fact, sometimes take very big transfers of pathways. And I think that good examples of that from genetic history, biotechnology history, is that E. coli is a reasonably small genome organism, and a lot has been done with engineering E. coli.
Right. So I know in terrestrial agriculture, one of the big concerns, I mean, the things that farmers fight all the time are crop protection. How do I keep pests and pathogens from eating my crop or a fungus from taking it down? Will that be the same in a biofuel crop of cyanobacteria?
Oh, absolutely. So anything that an organism is making that we find valuable is probably going to have a good source of carbon in it. And carbon is what organisms use as the skeleton of everything that they need for their cells, which means that if there's something out there that is alive, something else is going to eat it.
That's right. So
it pretty much comes down to that. And so we do have to worry about invading organisms of a couple of sorts, either that will compete for nutrients and compete for resources, or that will actually consume and otherwise kill the crop.
So weeds versus predators or pathogens.
Exactly. And there are many of both kinds that are out there constantly moving around and settling anywhere, including in the ponds.
So what are some of the strategies that your group is thinking about in order to give crop protection to these?
Well, for different kinds of algal crops, there are different kind of characteristically notorious pests. And for cyanobacteria, if you talk to people who are raising these ponds, they'll tell you that amoebae are a big problem. Now, there are lots of different kinds of amoebae, but knowing that amoebae are a problem, one of the things that we've done is, along with our collaborators at the Scripps Institution of Oceanography, we've been identifying amoebae that specifically eat the cyanobacterium that we use as a model, for which we do have a lot of genetic tools. And once we had that predator-prey system set up, we could screen an entire library of mutants, which means a collection of mutants where each one is missing a different gene. And we were able to find that we could knock out a particular pathway that affects the cell surface, and the amoeba no longer then, this particular amoeba anyway, no longer eats our cyanobacterium. So that really gave us a first clue of one of the things that we can do to make cells resistant to amoeba, put some kind of a mask on them so they no longer look so tasty. And there are probably many other ways, but once we have the question, we can go in and ask it. And
so it sounds, though, that crop protection in algal biofuels will be similar, not identical, but similar to the strategies we've employed for terrestrial crops.
You have to identify the pests that you're concerned about and try to find a solution that works with that pest, and then you have to go back and ask, well, how general is it, and does this protect from other pests, or do we need some other strategy for other pests?
Okay. I know one of the other concerns in any biofuel, in any crop, and especially if we're going to use crops like cyanobacteria for fuels, are both the water utilization as well as the nutrient utilization. Now, you mentioned that nitrogen can be fixed by these, but what are some of the other nutrients, and what are some of the other strategies that we might use to make sure that we're efficiently using those?
Well, phosphorus is another one that's quite important in biological systems, and phosphorus is an issue really for all agriculture and will certainly be an issue for algal crops. Right. And recycling the nutrients is an important avenue going forward to make sure that the phosphorus in the water gets reutilized, that any of the biological material, spent biological material that the fuel molecules have been removed from, that gets put back into the system so that we can recycle those nutrients.
And in the nitrogen fixing then, how is it that cyanobacteria can do this where most terrestrial plants and certainly eukaryotic green algae cannot?
There is, cyanobacteria aren't the only organisms that can fix nitrogen, but most organisms don't, and the ones that we do know of are prokaryotic, although sometimes they live in symbiosis with eukaryotes. Right. And nitrogen fixation requires really a lot of genes and to make not only a lot of enzymes, but to specialize the cells to be able to keep the oxygen tension low. So the enzyme that's used to fix nitrogen from the air into ammonia is very sensitive to oxygen. And one of the things that we see in these species that can is that they have a number of different strategies for having their nitrogenase enzyme active in some sort of an intracellular environment where the oxygen tension is very low.
Right. Hence the specialized cells that you showed in that one. Okay, so you know, we talked about the potential for biofuels. You showed in one of your early slides that cyanobacteria can be eaten actually as a food. So obviously we can make food and fuel out of cyanobacteria. What are the other potentials that you see in this that sort of excite you about the future and where the field may go?
Well, I think that it's very important that we learn to replace petroleum as a source for all kinds of chemicals. So it's not only a matter of fuel or of food or medicines, but also just a lot of the everyday things that we use that are currently made of plastics. And so I think as a source of the carbon skeletons that can be used for a lot of the materials and the structures that we need in our everyday life, the ability to use algae, both cyanobacteria and eukaryotic algae, as a source of the building blocks of those materials will be very important.
Okay, great. Well, thanks very much for the introduction to cyanobacteria, and best of luck with your research.
Thank you.